Hello, this is Kevin Edwards from Illinois State University, and today I'd like to talk to you about some specialized imaging modes that are available on our confocal microscope. This facility is open to regional users, so if you're interested, please contact me at this email. Now, if you would just like to learn some more about the scope, you can check out our facility website here. Here you'll find the specifications for the system. Here you can find some videos on the facility that I posted to YouTube. I won't try and duplicate this information here today because you can find it here. But there's an overview of the facility and some of the background information on the imaging modes that we'll talk about today. Instead, today I want to show you some updated examples of how these different applications can be used for various research questions. And so the modes I'd like to discuss today are lightning adaptive deconvolution, spectral fingerprinting using Lambda-lambda plots, and real-time FLIM, fluorescence lifetime imaging. So just briefly, I'll mention the facility is a Leica SP8 Confocal. It's mounted on an inverted microscope, has an excellent range of different objectives available, and the confocal features a 5-detector array with 2 photomultiplier tubes and 3 hybrid detectors. The hybrid detectors are very low noise, very high sensitivity. Now, the first mode we'll talk about is spectral fingerprinting, and this relies on 2 different features of the system. One is that it uses a white light laser for excitation, and this allows you to choose any wavelength from 470 to 670 nanometers and send that to the sample. Then, using the Leica prism system for a detection, we can pick up any wavelength range from the blue out to the far red. Now, in this experiment, we stained a Drosophila salivary gland with 2 different lectins, each labeled with its own Alexa dye. We also stained with acridine orange, which should provide orange fluorescence when it's bound to RNA and green fluorescence when it's bound to DNA. So we wanted to see if acridine orange would be compatible with these 2 red Alexa dyes. So to do this, we're going to take not just a single image, but actually over 1,300 different images, each one at a different excitation emission combination that can be achieved with the white light laser. And so every pixel in the image actually has 1,300 different intensity values underlying that pixel. So how do we show that information? So we can use a heat map where we're plotting every excitation versus every emission. And when we do this, we can see each of the dyes. Here are the 2 Alexa dyes in the red and far red, and they're producing fluorescence in their expected range of excitation and emissions. Here we're looking at a specific area of the sample that has all 3 dyes roughly in equal abundance. But if we look over here and focus in on a glue granule, we see that that particular structure is only stained with the PNA lectin and its red Alexa dye, and the other stain is not present there. So we can zero in on individual structures or areas of the image and figure out exactly which dyes are present. So what about acridine orange? Well, here we see that the structure of the fluorescence looks quite different than the Alexa dyes. Acridine orange has multiple peaks of excitation. However, all of its emissions are in the green range. Well, so what about the orange component? Well, it should be out here, but it seems to be absent from this experiment. So this is a good reminder that your dyes don't always act exactly as described in the literature. And actually, it's a good idea to check the behavior of your dyes in your sample. So in this case, we might not be seeing the orange fluorescence due to the concentration of the dye or the pH or other factors in the protocol. Now, the second mode to talk about here is FLIM, or fluorescence lifetime imaging. In this mode, we're ignoring the spectral information, so we don't have excitation emission data. Instead, what we're going to measure is how fast a photon is emitted after the dye becomes excited. So we get the timing between the excitation photon hitting the dye and the emission photon leaving the dye. Those timings are a collection of values which can be plotted to give you a summary statistic, which is tau, and then tau can be color-coded. Tau will be characteristic of each different dye, and in fact it may vary for a given dye under different chemical conditions. Now, in this example of FLIM, we've taken a fly head, decapitated and placed in halocarbon oil. So we're looking at a live sample, no fix and no stain. So what you're seeing is purely autofluorescence. we've opened up the spectral range. So there's really no spectral information here at all. We're looking at all the autofluorescence from the cuticle of this structure. This is actually not Drosophila, this is a phorid fly. We initially wanted to look at the eyes and determine if there was autofluorescence there. There wasn't much happening in the eyes, but when we saw the antennas, we saw a whole range of different tau values, different lifetime values, showing up in the antenna. Now again, this is all autofluorescence of the cuticle, and so therefore the different colors indicate different chemical composition of the cuticle in each of these different structures. In particular, we see that each morphological type of sensillum has its own characteristic tau value. So here we see these longer pegs have one value, the shorter pegs that have a different value, and they're both different than the longer hairs. So it appears that the cuticle can then be tailored to suit the needs of different sensilla. That's an observation that would be kind of hard to make using other methods, and it seems to be novel, at least I haven't seen it elsewhere in the literature. Now stepping back to Drosophila, here we see a developing embryo that is non-decoriated, so it still has the eggshell. This embryo is expressing GFP beta-catenin, which is a junctional marker, which you can see here in the amnioserosa and then here in the epidermis. Now, a non-decoriated embryo presents some imaging challenges. The chorion is very optically complex. It's also very autofluorescent. Now, here's the intensity image, but here if we switch on FLIM, we can see that the different components separate out very nicely. GFP has a very distinctive lifetime value, and if we just zoom in on the GFP signal here, we see this peak. That corresponds with the known lifetime value for GFP between 2 and 3 nanoseconds. Meanwhile, if we look over here in the chorion, we see that the chorion autofluorescence has a much shorter lifetime. And in fact, we see two different colors. So we look at the main body chorion here, the lifetime value is between 1 and 2, but the region in the dorsal appendages actually has a much shorter lifetime down here. So there are chemically different types of chorion in the eggshell, and they're both distinct from fluorescent proteins. Right, so the last mode I wanted to talk about today is adaptive deconvolution. This is branded as lightning in the Leica system. In lightning, the successive images of a Z-stack are compared to each other to try and reassign intensity information from where it's collected to its point of origin. And this tends to very much tighten up and sharpen up the representation of objects in your sample. It also reduces background haze and essentially eliminates noise in the sample. Now, we've been using lightning pretty extensively over the last couple of years, so I wanted to give you my impression of the pros and cons of this method using some apples-to-apples comparisons of standard imaging with lightning imaging. So in each of these three examples that we'll look at, what I've done is to take an image of an M-cherry labeled fusion protein along with other markers for different features of the cell. And I've done a Z-stack in lightning, and then I've done the same Z-stack with standard imaging using the same resolution, the same pixel size. Now, when you do lightning, the pixel size is greatly reduced compared to what you would typically use for standard imaging. So for standard imaging, you might go for a pixel size of something like 50 to 100 nanometers. But with lightning, the program is set to choose a pixel size of about 30 nanometers. So you get significantly finer resolution of whatever it is that you're collecting. Now, this would be empty magnification in standard imaging. But with the lightning image, you're getting information down to that level of about 100 nanometers. So you really do need to use 30 nanometer pixels. So here we're looking at a fusion protein that makes structures inside the nucleus, and that's shown in green, and DAPI is shown in red. So if we zoom in on part of this nucleus, we can see that there are features here in red and green that lie adjacent to each other, and yet they're very clearly resolved using lightning. Now remember that the pixel size here is 32 nanometers, and we're zoomed in here to see individual pixels. We can see that these two features are separated over a distance of only about 3 pixels. That's about 100 nanometers. In contrast, if you image the same volume with the same pixel size with standard imaging, those two objects blur together and can't really be resolved. Now in this example, we're looking at a fusion protein that makes aggregates in the cytosol. Those aggregates go from below the detection limit up to about 2 microns in size. Here we're looking at the same set of aggregates with standard imaging and with lightning. You'll notice that the edges of the aggregates are very blurred and indistinct with standard imaging, and they're very sharp with lightning. So only with lightning can you get a really accurate representation of the shapes of these aggregates. Now, to get a sense of the lateral resolution that we're achieving here, I looked at the smallest aggregates, and we can assume that the real size of those structures is smaller than the resolution limit that we could achieve with either mode. Now, those features with lightning present as about 3 to 4 pixels wide. That equates to about 110 nanometers. The same features with standard imaging present as about 5 to 6 pixels, or roughly 180 nanometers wide. So that indicates that lightning gives us a very significant increase in lateral resolution. Now over in these panels, we can see that there's also an improvement in the Z resolution when we use lightning. Part of this occurs at the collection step. When we collect a lightning Z series, we set the pinhole very narrow to exclude out of focus light. So here the pinhole is set at 0.68 area units. Now for a fairer comparison, we also close down the pinhole for the standard collection. In this case, we use 0.9 area. So both of these are collected with very good Z resolution. However, in the stack, you can still see that there's quite a bit of out of focus light present in the standard image. Here in the standard image, we're looking at actin cables in white and our fusion protein in green, and we can see that the actin cables, even though they lie at a different focal plane above our aggregates, the image of those actin cables is very bright and it's intruding into our image of our fusion protein. In this cell, you can see some out of focus actin cables, and they've been almost completely removed by lightning, so that our protein of interest is no longer obscured. Now, in contrast to the aggregates we saw in the previous example, in this last example, we'll look at some more extended filamentous structures. So here we're looking down on the apical surface of the follicle cell epithelium of a fly egg chamber. And one of these cells is expressing an unknown fusion protein that makes a pattern. This pattern is confined just to the apical surface, and the protein seems to exist right at the same focal plane as the microvilli, which you can see here with actin. In both cases, those structures are much more well defined with lightning than with standard imaging. And with that better resolution for both structures, that makes the colocalization image a lot more informative than it is here with the standard image. However, you can also see a disadvantage of lightning in this image. So as you go through the deconvolution process, the computer makes a call and decides what's background versus what's signal, and it eliminates the background. So here in the standard image, we can see 2 cells adjacent to this high expressing cell. They're expressing our protein at a low level. So we can easily see this in the standard image, but here in the lightning image, that lower level signal has been treated as background and eliminated. So we are throwing away a bit of data there. Now, given the pros and cons of lightning, I would suggest that if you publish lightning images, you disclose the use of lightning and also provide the non-deconvolved images as a supplement. So overall, I think that lightning is a very powerful method for looking at well-defined objects in the range of about 100 nanometers up to a micron. So if you're interested in giving it a try, give me a call. I can either advise or you can come and try it on our system. All right, thanks very much and have a good day.